The setting outside is moody, and I gotta complete my science stuff. Fuck. Yeah. So this is the amino acid worksheet. I have just managed to complete it recently. Bro. Oh my god. Okay, you know what? No. It says it right here. It says it right here. I just fucked it up. Okay. I thought I did this unnecessarily. Doesn't want to give it to me. Okay, so yeah. Label the on each amino acid. So basically I just took a screenshot and just circled all of them on one note. So yeah, that's what I did. It's pretty easy. Here's all the labeling. And I said that all of them look to me like D, because if we were to go back into the textbook, hang on, scroll out, scroll in. Okay, so basically, if we were to draw like a pyramid, and you would imagine a note. Okay, so how does it describe it? Now, where's the textbook? Okay, so yeah, D is blue, red, green. Okay. Red, blue, green. Okay, just ignore the fact that it's like that. Okay. So this would get number 1 priority all the time because it's a nitrogen. It's a different guy. This would get 2nd priority because it's got 2 oxygens. And then this would just be an R group. And usually this is like C, C, C. And then usually then comes like the R component of it. If you were to go here, you would see that mostly being the case. And so that's why I would give the priority to this. Also, it's double bonded to an oxygen. So, for those reasons, I labeled it as 1, 2, and 3. And just to avoid any confusion, all this R, and this will be C, this will be Kun. Okay, so basically, because 1, 2, and 3, 1, 2, and no, 1, 2, and 3. Okay, so the way I drew it, no, I fucked it up. Nope, that's my bad. I fucked it up. I fucked up the colors. Okay, so this is 1 because it's the nitrogen group. This is 3 because it's the R group. Yeah, I messed up the colors. That's my bad. Okay, so essentially 1, 2, and then 3. And because it rotated like in that direction, like this, as you can see here, you can do the same thing here. Hydrogen doesn't matter. You would give it an L, okay? And the opposite, like if we were to switch these two, it would be D. And so when you scroll up, you would see none of them follow that structure, okay? So 1, 2, 3. In fact, it would spin this way, and so it would give a D conformation. 1, 2, 3. In fact, it follows that perfectly. And so this is why I did that. I just went too much into detail. I'm going nuts. Okay, so yeah, that's literally given in the textbook right here. This is just me fucking around. Show a peptide bond, label everything in the peptide bond, and like I label everything. I forgot the water initially. Okay. Have both hydrophilic and hydrophobic properties in the side chain. I don't know what that means, but whatever. Okay. Polypeptide, not folded, folded in correct shape protein. Okay. Similarities, I didn't go too much into detail. Yeah, but basically this is all it is. And here I just highlighted some of the best things. And then this one I just underlined because that's gonna look bad, but you know what, whatever. We do things the way we do things. So this is basically the main points I want to get across for this, just in case I ever want to come back to this. Yeah. So basically this is the polypeptide chain, and then this would start folding the way it showed. Come on. In the textbook. But it won't necessarily do this. This is just an example to demonstrate, which is possibly the best example that I used. But it'll do one of these two things. I just love this picture. If I had to highlight this chapter, it'd be this picture. So yeah, that's basically it. This was a nightmare. I didn't know what to do, so I did this, thinking that I had to somewhat bend it, but all that was basically the peptide bond would do the turning. So it's because of the peptide bond, it would turn. But I don't know. But here's the thing: it did 2 turns. Like it turned once, and then it turned a second time, 3, then 4. And so because of this, every peptide bond has 2 turns, and so that's why I would say that this had 44, because yeah, and between each and every one of them is a peptide bond, and it would turn twice. So if we were to go back to the textbook, yeah, right here. So you would see, let's just assume going this way, 1, then 2. Otherwise, it would just be like a straight V. But instead, it does more like a W, like a repeating W. So like, yeah, so there's 1, then 2, and then it goes back in that same direction. 1, 2, and then yeah, it keeps doing that. And so that's why I justified it as saying this. And I just realized this now, as I was saying it out loud. Okay. And then this is just simply like, if one of these was like 0.5 and we put like 23 of them together, it's just basic math. Okay, this, I thought it was like AL, like the amino acid. Turns out the TA got lazy and did this for, what? The amino acid. I don't know if it's a TA that wrote this or the prof that wrote this, but this was hilarious. This cracked me up so hard. So yeah, this is what I did. Yeah, this confused me a lot, but basically what this is saying is like, what would be in the center? That's what this is, but member anchoring was a little bit confusing. I don't know if there's any like, maybe special cases, but I'm not too sure. Might mess up the chain. But yeah, that's basically it. This is what I have to say for the thing. And then 10 is just basically is reversible pros, blah blah blah. I just copy pasted this from the text. Protein. And yeah, that's basically it. I'll probably post a link of me doing this for both the nucleic acid and the amino acids. But yeah, this is about it. This is all the homework. Pause anywhere that you need to gather this information. And you're good to go. Okay, so moving on. Okay, the phosphate group. All 3 make up the new building blocks of DNA. We'll get into that later. But what makes it up specifically? Phosphate group, 5-ring sugar, and nucleotide base. Okay, so basically, you had to draw everything out and like say the similarities and differences and point out the differences. Where is it? All the parts. Numbering all the atoms. You had to number them as well? You know what? Fuck it. I don't got time for this. Yeah, it's like a 1 hour, 1 hour for me. Fuck that. So yeah, just copy paste. It looks generic, same. Okay, but yeah, for U and then T, it's different because the methylation gone, and then the OH in the second carbon is also gone, so it's ribose deoxide. I got paranoid. I should have labeled it, but I didn't. But fuck that. There's the draw. There's the number, possibly wherever, circle, whatever's involved. And blah 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 blah. There's some like wordy answers that you had to put in here. I said fuck it and put all of them in the same goddamn picture because like I got lazy. Okay, I got lazy. So yeah, 3 is the same. So wherever there was like thing I could just draw in here, I just drew it. So like the diester diester bonds with the phosphate group. And then so it said towards the end that you had to add like this specific thing. And so this is just basically what it is, but guanine instead. So you had to make that apparently very clear. And so yeah, I had to justify the bonding, attaches on the 3 end, everything makes sense. Okay, so draw a complementary strand, label all the things. So basically you had to do this. I said fuck it. I took this, flipped it, and then flipped it again, okay? But the thing is, I had to redraw like the NHs because like I compressed them here, but then it came back to bite me when drawing the hydrogen bonds. And also for CG, you're gonna have to flip the ketone and the nitrogen group. It's not like your fault in drawing, it's just that I tried this with the other thing. It did not work. So you're gonna have to do that. And you're gonna have to draw the bonds, label. I just left some of this in there just because I was paranoid. So hydrogen bonding all of them. Yeah, I think it's hybridization. It's a technical word, but I'm too lazy to change it back. Too much effort went into this. I didn't fully understand this, so I just put in this, and then just relabeled it like that. So the coding strand and then the template strand. So the template is, I believe, this, and then this is the coding strand. You know what? I should make that clear. Template strand coding strand. Okay. Okay, so why is there no TPP in cells? It's the alternative. DTTP, it simply exists as. So that's the simple reason. So TTT will never exist. I say never exist, but that's my limited understanding. Okay, so this is just my answer. This is how I understood it. Okay, so why is there uracil? Okay, so basically I had two reasons for this. DNA polymerase recognizes DTP as the building blocks of DNA. So yeah, that's basically what I did. And yeah, just you can pause, you can read that. Okay. So this one was just simply, it tried to be confusing, but I don't know, man. Okay, so there were two rules. So the first rule is that it equals to this, and the second rule is, where's the two rules? This is the second rule, and then the first rule was that the double-stranded of the DNA had to be this. This was the second rule. Okay, that's all you need to know. So this was just basic-ass math, assuming 100% is the max. So that's how I figured it out. So 100% minus 56, that's 44%. You know what? I gotta show my calculations. Because they might be petty and take out marks. Okay, I have not been strict with my work. There are some places you could take out marks, but that's if you're extremely petty. Don't be petty, whoever's marking this. Okay, the DNA 20%. So yeah, basically just the equal CG 20% plus 20% equals. I love doing it like this because I understand what I'm doing. Okay, so 100% minus 40% is 60% for CG. No, it's actually for AT? Yeah, it's for AT because we already know what C is. Shut the fuck up! Okay, so why does RNA not obey? Because as stated in Rule 1 and Rule 2, this rule can only be applied to DNA with CG-AT. What's with you? Youth, grammar Nazi. PH. Okay. And then RNA uses U instead of T and simply doesn't work. And so this fucks up the rule that C is equal to T. There's more complicated, but I don't know. So yeah, that's simply the ins and outs of it. And that's sort of the only justification I could find for it. If one strand of A was found... Okay, this is simple, okay? So basically, bacteria A would be expected to have more CG like what? In DNA, like more frequency than bacteria B because it would stood a higher temperature because 3 hydrogen bonds are harder to break. And the alternative is 2 hydrogen bonds. And so, because there's so many more hydrogen bonds to break, like, it's like the stick example, all right? You can break one stick easy, but not like 1,000 sticks all at the same time. So it's going to take time. And so that's why bacteria A in my eyes could withstand it. Some of them go blue, some of them go black. I don't have the time to change it. Just fuck that. Important thing about the major groups and minor groups is that it helps proteins. Okay, so that's like the technical reason, but is the space between entwined to the grooves? You can basically pause and read that. I just came up with some bullshit. Okay, so it's basically all you need to know is that spaces between the grooves of different widths. So that's what I would highlight as the major thing. I just basically copy pasted that, and yeah, this video will be linked. Like the rest of this will be linked to show you what I see. But that's it. That's the end of it. Thanks for listening. Bye-bye.